My name is Zachary Hoyer, and I'll be presenting a paper called Glucocorticoid Exposure During Hippocampal Neurogenesis Primes Future Stress Response by Inducing Changes in DNA Methylation. And these are the authors, and this is the citation if you want to check it out. To begin my presentation, I want to go over the stress response. So, what will happen first is you will get this information sent from different regions in your brain, and it'll all get filtered by the hypothalamus, more specifically the paraventricular nucleus. And all this information will get filtered and assessed, and if it decides that you need to have a fight or flight response, it will release this corticotropin releasing factor, and it will talk to the pituitary, and this hormone will cause it to release corticotropin. And this release of corticotropin then acts on the adrenal glands inside of the kidneys, or on top of the kidneys. And this hormone will now release glucocorticoids and your epinephrine and norepinephrine. That'll cause the more physiological, like instant response. And then you'll also get this release of glucocorticoids. Glucocorticoids are pretty much the stress hormones. Right here is the structure of cortisol in humans, and it is what is released that causes a more long-term effect on the human body. It can help sustain some processes, so you have the energy to keep up the stress response, and it can also, it's like also partially a neurohormone. And here, this kind of cortisol analog, you can see the fluorine right here, the lack of a double bond over here, lack of a methyl group, is called dexamethasone. And it is a synthetic stress hormone that we use in research to help kind of mimic cortisol without actually using cortisol. And it'll be used pretty heavily in this paper. And these hormones can work on things called GREs, or glucocorticoid response elements. Now, these GREs are expressed in pretty much every cell type in the body. And if it gets acted on by cortisol or this dexamethasone, it will cause an intracellular response, a cascade, like a second messenger cascade. And actually, I don't think it will cause a second messenger cascade. It will essentially just act on these GREs, and these GREs will go into the nucleus, and they will either upregulate or downregulate transcription of DNA. They also have effects on methylation of DNA, and yeah, that's something that will be talked about in this paper. Since stress is present in pretty much everyday life, one question is, how are glucocorticoids regulated during pregnancy? Because pregnancy is a very stressful thing, very hard on the body, and it is kind of questioned how it is regulated and how it might affect the growing fetus. And so this here is a little diagram showing one of the ways that stress hormones or glucocorticoids are regulated. Now, this is in a rat model because it is corticosterone and 11-dehydrocorticosterone. But in humans, corticosterone would be cortisol, and this one would be cortisone. So basically, what happens is during pregnancy, the inside of the blood of like fetal blood, there will be a production of this enzyme called 11-beta hydroxysteroid dehydrogenase, and there's a first version, that's the reductase version, and a second version, that's the dehydrogenase version. So reductase version is just adding on a hydrogen onto the inactive form, and so you're going to use up NADPH, and then the dehydrogenase is basically taking off a hydrogen from the active form and adding it onto NAD or NADPH, depending on where it is in the cell or outside of the cell. And basically what happens is this enzyme is produced in mass amounts, and it will deactivate cortisol or corticosteroid in mouse models, and it'll turn it into this inactive form that cannot create a stress response. And so this is one of the mechanisms that are employed that help regulate the amount of stress that can actually reach a fetus. One question that is asked is if the stress from a person can reach a fetus inside of them, a big question is there a link between that stress and the fetus's development? And so here's a little diagram that is going through the development of a person. So it goes prenatal, postnatal, adolescent, adulthood, and then like late adulthood. And so what you can see here are these dashed blue lines kind of are representing this developmental region. Not a lot is known about these regions just yet, but that's where all the research is kind of gearing towards right now. And it's not necessarily understanding where stress plays a role in these mechanisms. And so one thing that it does kind of show here, though, is postnatal. These big blue lines are the most critical points for these regions in the brain. They're like most critical growth periods, or almost, or like where stress and glucocorticoids will have the largest response. Here, it's kind of debated in these dashed blue lines. But if they are hit in these quote unquote critical periods, it will have a large effect and it can be pretty detrimental, especially over here. And as you get older, stress will decrease or kind of degrade these regions in the brain more. And so what we want to focus on in this paper is the hippocampus. And what they're trying to elucidate is whether or not these glucocorticoids have as large an effect that they could. They want to kind of find out this link and just how pertinent it is and whether we really need to be on the lookout for stress in the first, second, third trimester. To help find out whether or not stress has an effect on the development of the hippocampus and other regions in the brain, what this study is going to focus on is hippocampal neurogenesis. So what is hippocampal neurogenesis? Well, what you have is you kind of have these stem cells, and they will proliferate and they will turn into activated neural stem cells, and this is all occurring in the dentate gyrus. And these neural stem cells can split, and they can make cells that want to, like these neural stem cells can just kind of split continuously. They can keep keeping on. So they will just continue splitting. They'll just keep doing like proliferating and everything. But if a cell wants to turn into a neuron, it's going to have to differentiate. And so you'll get this activated neural stem cell, and it will have some chemical response that changes the genes inside of it, helps kick on the proteins that are needed for it to be a neural progenitor. And once it makes that switch, it won't go back to the activated neural stem cell. It will stay a neural progenitor and go through this process known as neurogenesis. And so it's going to differentiate. And as it differentiates, it's going to become whatever it wants to. It can become a glial cell, it can become a neuron, basically just anything. And then it's going to migrate, and then it's going to integrate, and that's kind of the process of neurogenesis that can go on inside of the hippocampus. Now, as this goes on in the hippocampus, we know that hippocampal neurogenesis is related to a lot of learning, and if there's not a lot of neurogenesis, you'll be able to remember a lot, but you won't be able to learn stuff as well. And if you have more neurogenesis, it's kind of shown that you can have an easier time learning, but you don't have as solid of memory. And so this hippocampal neurogenesis is a type of plasticity inside of the brain, how it can cause long-term functional changes, functional or structural. And some of the ways that it actually goes through this, I talked about it a little bit while going over neurogenesis, is something called methylation of DNA. And so what can happen is you get these they're called GPC sites or CPG sites, if you want to be more consistent. And if these are methylated, they will either decrease or increase transcription, but usually it's a decrease in transcription of this DNA. You can also have methylation of just a CG site, but a lot of enzymes specifically target CG sites. And so methylation is far more likely in CG sites. And it will happen on the 5 carbon of the cytosine. And yeah, it doesn't really happen in any of the other nucleobases. It can happen sometimes on the phosphate backbone, but that's more about histone regulation. That's much more rare for any of that. Well, not phosphate backbone, but the tails of histones. And these are basically just ways that you can have inheritable changes in gene expression. And so it will either increase or decrease gene expression of whatever gene it's in. And these changes are just a way for you to have an epigenetic mechanism as you live your life and you build experiences, and you need certain proteins just by the way you've lived your life. Go over the goal of this paper. It is to examine how glucocorticoid exposure at different stages, including proliferation, differentiation, and post-differentiation, affects DNA methylation and whether these changes are persistent. So, as I showed in this figure earlier, we are studying this prenatal stress, this area right here, and a little bit of the postnatal stress, and to see if it has a long-term effect on this prenatal organism. To actually begin this research, these researchers needed a system that they could perform tests on in a humane way and an accurate way. So the best thing that they could come up with was these hippocampal progenitor cells, or HPCs. And they used this instead of like an animal model or, I guess a fetus or something like that, because these are the most accurate and representative cells that you can get of a growing fetus. And what they found out is that these cells represented about the second trimester of a pregnancy. And so these cells were right at the stage of whenever the hippocampus was going through a decent amount of neurogenesis. And so they did 4 different tests, 4 different cell lines to represent when stress, or DES, this glucocorticoid analog. They did 4 different cell lines to represent when this stressor would be introduced to a fetus. And so this first one here is the pro cell line. And this means basically DEX was introduced for 3 days during the proliferation phase of these HPCs, and then the DNA and RNA was extracted from these cells using nucleases, well, not nucleases, but basically just using drugs to lysate the cells, get the DNA and RNA out, and then they performed Illumina sequencing, which is basically just a Sanger sequencing or a more advanced Sanger sequencing. And so that's the proliferation cell line. This is the pro-diff cell line, and it basically means they introduced DEX for the proliferation phase and the differentiation phase. So about 10 days total. And then they extracted the DNA, RNA, and then they did the proliferation, differentiation, and washout phase. So they introduced DEX for proliferation and differentiation, and then they let these cells sit for 20 days to see the longer term effects that the stressor would have on these genomes and these proteins expressed by these cells. And so this fourth cell line, they didn't introduce any DEX into the proliferation or differentiation phase, just 10 days after those two phases. So the cell is already differentiated, it knows what it is, and then they started introducing the stressor, and then they had that same 20-day washout, and then the extraction. And they also did another set, another 4 cell lines where they did the same things, except instead of DEX, they did vehicle. So basically just saline or some other drug that they decided to use that wouldn't affect or would act as a control. Figure 1b and 1c, and it's the effects of DEX on both gene expression and methylation. This is figure A. This was from the last slide, but I'm just showing it again because it acts as a key almost. So as you see these 4 columns, they are associated with each of these colors, and they are associated together. But here in B, we have the effects of DEX and vehicle. We're not showing the vehicle effects in this picture, but it is showing the effects of DEX on 1 differentially expressed probes and comparing that to genes. And so what they're doing is they're testing to see like this change in expression. So the change, and then they kind of subdivide it into downregulated and upregulated. That's a little less important right now, but they're just showing this change right here. And as you can see, for protein expression, you see a bigger change in the counts of the differentially expressed probes and the genes. So having more of these probes expressed means that this DEX is having an effect in these cells. And the same is said, this is for the pro cell line, so just DEX during proliferation phase. And here you see the pro diff. You still have a change, not quite as much as the pro cell line. And then here you have the pro diff plus wash and the post wash. So almost no change with the post plus wash, and very little with the pro diff plus wash, which is very surprising. And then in this line you see methylation. So surprisingly or unsurprisingly, you'll see more differentially methylated sites for the pro cell line, but then surprisingly, you don't see very much change in the methylation of the pro diff sites. There's almost no change at all. And so you do see a slight increase with pro diff plus wash for the amount of methylated sites that you see compared to the normal genes. And then about the same thing for post plus wash. We have an analysis of the data that's been received so far, and it's called GAPS, or gene activity in pattern sets. And this analysis was done on the differentially methylated sites. So this is a type of a transcription factor alongside the glucocorticoid response elements. And it is comparing the vehicle to DEX. So these bold lines are the average of this number of genes, and in total we have about 500 genes here. And so we'll talk about the average in a second. Actually, these are each 4 different sets of genes, or 4 different trajectories that this number of genes takes, and they're roughly the same. So they all kind of do kind of similar things, and they can be grouped up in a way where, oh, these genes are having, like, these genes are being affected by something in the same way. So in this first trajectory, it's called trajectory green, we have about 71 different genes in here. And basically, with the addition of stress, these genes are reacting the same way. GAPS is an algorithm that will take all the information and it will compare different genes, and if they're multiply regulated, so they have a bunch of different transcription factors, it will sort all of that out. And if we're talking about the effect of stress on these genes, all 71 of these genes work roughly the same way, and these bold lines represent the averages. So trajectory green, they're all kind of having this descending slope, which in this case represents a decrease in methylation, as seen by this first kind of column right here in column C. And in this column, or the second C column, it's representing the expression of these genes. And so trajectory green, you kind of see a decrease. Trajectory red, you see a little bit of an increase. And then for trajectory blue and beige, it stays roughly the same for all of them. And this trajectory, it's just called that because with this study, the way that they set it up, you can almost get a timeline of neurogenesis. So here you have the proliferation phase, proliferation differentiation phase, pro-diff plus wash, and then post-wash. And they weighted all of these differently, so it should make a general type of timeline. I don't exactly understand how it works, but these trajectories pretty much just show the where the methylation and gene expression is going inside of these cells, and that these inside of these, like, these genes, and it's basically just clumping these genes together into kind of like how they are all affected. And this is figure 3b, gene response to a secondary acute stressor. This is a very interesting graph because it's a violin graph, which are very pretty. But it's basically saying that in a secondary acute stressor will cause a greater effect inside of the genome and on the genome. So here we have the fold change, so basically just how much change the system has gone through. And this is the pro plus differentiation plus wash, and then 20 days after, so the wash, they harvest and extract RNA and DNA. And right here, it's about 1, 1, and 1. So basically almost no change in what they are experiencing. Like, this is basically the baseline that they've set, is this pro-diff plus wash, the change in gene expression and methylation. And so here is the vehicle plus an acute stressor. So they weren't given any of the DEX. And so here is kind of the control group. And then here's pro-diff plus wash plus the acute stressor. So they're comparing this to the control and also the control pro-diff plus wash. And here you can see that it just kind of shoots up. You get huge fold chains or huge fold, like, amount change in the gene expression. And so this is just basically saying that in a secondary acute stressor will cause a great effect on the actual genes, either methylation or transcription, whatever is being measured as the response, that you will see a much bigger change. And here it's showing methylation. Finally, one of the last things that this study decided to test was they tested to see if there was an overlap in the genes that are being expressed expressed in these hippocampal progenitor cells and the umbilical cord blood that are having these longer lasting differentially methylated this basically just being methylated. So they're trying to see if there's any overlap in the cells that had long-lasting methylation changes in this study. And in a database that, or I guess a study that was done about umbilical cord blood and the methylation or the methylated genes. And so they wanted to see if these epigenetics had any overlap, and about 500 genes did. And so what they did is they plotted they did more statistics on the overlap, and they basically rated the polyepigenetic score. So if it had more methylation, or I guess no, less methylation in these genes, it was given a negative value. If it had more, it was given a positive value. And what they found is in CESD, or this depression test, that if you ranked lower on the depression test or higher on the depression test, it translated to a lower polyepigenetic score. So that was less methylation and a basically an increase in your chances of, well, just basically less methylation and an increase in transcription of those genes that are associated with anxiety and depression later on in life. And so what they found is in the person who was pregnant with these fetuses, that they had, if they scored higher on these tests to represent anxiety and depression, they most likely had a lower polyepigenetic score, meaning that the fetuses also had a higher chance of having those same types of conditions later on in life. Take-homes that I have for this study is glucocorticoids do lead to lasting changes postnatally, but few relating to long-term gene expression. So there's no real long-term protein like elevation because a fetus was exposed to a increased amount of glucocorticoids, but it does cause long-term like methylation changes inside of the fetus, which will open the door for future stress responses to be more severe. And this link between the maternal stress and depression may be caused by the shared GP or CPG methylation sites, but more research needs to be done on that. So essentially, having a more depressed or anxious pregnant individual can have these long-term epigenetic effects on like hippocampal gene expression, and that's something that hasn't really been shown before. So it's very, very interesting. And basically, how do you think stress could be mitigated during the prenatal developmental period? That's something that will be coming up, or has always been a question and will continue to be. It's a very important one for the development of a child. And then, do you think this is an evolutionary adaptation, or it came about due to the finicky nature of the shared umbilical cord? Thank you so much. Have a great day.